Okay, so for today, the lessons, these lessons, we're going to look at about transport, and also, as opposed to what we have learned in the diffusion, facile diffusions, osmosis, active transport, indirect active transport. Those we take in the particle one by one. Are you clear? So they have to line up one by one, we take them in. Bulk transport basically means that we take them in a huge quantity or large quantity. Okay, for example, lights. Okay, protein. Protein is very large. So if you want to take them in or you want to send them out, you cannot send them part by part unless you break it down. Are you clear? Okay, like hormone. So this kind of the process involve what we call the endocytosis or exocytosis, classified under bulk transport. So bulk transport is a mechanism of transport of large quantity of material. It can be entire bacterial cells. It can be entire virus or proteins. Or we want to send them out as a huge quantity. I don't want to send them out one by one. Are you clear? Like hormone, don't want to send one by one. It's so slow. I want to send them actually in a huge quantity. Therefore, I need to do this bulk transport. And two kinds of bulk transport in our syllabus that we need to know: endocytosis and exocytosis. So these two, endocytosis. So what endo means take it in, endo, enter into. Exo, exit. So means that out of the cell. So again, endo means take it in, exo means send it out. And those both processes require ATP. So why we need to have ATP? Because we need to have this motor protein called kinesins and dienes. So kinesin and diene, they are motor proteins that couple with the ATP hydrolysis to move along the microtubules. Still remember, I do said before, for the organelles to move along or move within the cells, and also for vesicle to move, we do need to have the railway track. So who act as a railway track here? Microtubules. Okay, but the thing here is, if you have a railway track, you still need to have the wheel, right? You try to imagine the LRT, okay, or our train. Okay, without the wheel, you cannot move, right? So you have the motor and the wheel. Okay, so the wheel here is the kinesins and also the dienes. So it's a motor protein that actually bring the vesicle as well as the organelles to move. Okay, so kinesin move by hydrolyzing the ATP. So when I hydrolyze the ATP, so this one release the energy. So in the microtubules, they have the plus N, also have the minus N. Plus N and minus N. So this diene and kinesins, they have the ability to hydrolyze the ATP and then move them. So kinesin move this protein, okay, this motor protein, move the particles or move the vesicle organelles towards the plus end of the microtubule, and diene move to the minus end of the microtubule. Okay, so therefore the vesicle and organelle now can move into the correct direction. So it depends on what proteins. Okay, so look at this diagram. So microtubules, we have the positive ends and negative end. So it depends on which kind of motor protein. So different motor protein will move. So therefore, the vesicle organelles, it depends on their interactions. If you choose the wrong platform, definitely you go to the wrong directions. Clear or not? You try go to your train station, you need to go through the correct platform. So it means that you need to go into the correct train. So the vesicle have to bind to the correct protein first. So if you take this kinesin, then it's going to move to the positive end. Are you clear? If the diene, then it will move to the negative end or the minus ends of the microtubule. So do you need to memorize? The answer is no. Just need to know that the movement of vesicle, okay, during the exocytosis or endocytosis, so these processes require ATP. And how they move? They move along the microtubules. So you do not need to remember the kinesins proteins or dienes, okay, motor protein. But what we need to know in our syllabus, basically the movement of vesicle. For example, from Golgi apparatus form the Golgi vesicle, and the Golgi vesicle move toward the cell membrane and carry out exocytosis. So the movement of vesicle require ATP. ATP. And also for the vesicle that move, okay, after the endocytosis. After the endocytosis, then we actually form the endocytic vesicle, correct or not? So this vesicle have to move. So the movement of vesicle again require ATP, not energy, but ATP. So the hydrolysis of ATP make this kinesin protein change the shape as if they're going to move, right? Like the leg, right? Can you see that? Move along these microtubules. Okay, so one of the role of the microtubules for the movements, okay, of the vesicle and the organelles, okay, along these microtubules. Okay, so highlight this one, guys. The only thing that we need to know actually is this. The processes require ATP, okay, because this vesicle and organelles, they move along the microtubules. And we need the ATP so that this motor protein can carry the vesicle and the organelles move along this microtubule. Okay, so we start with the first one, endocytosis. Endocytosis, we can divide them into two, which are known as phagocytosis and pinocytosis. So phagocytosis is more like eating, cellular eating. Pinocytosis, something like drinking. Are you clear? Okay, phagocytosis means eating, pinocytosis means drinking. So it means that phagocytosis involves the taking in of the solid compound or solid particle. Pinocytosis taking liquid solutions. Okay, but both of them involve what? Engulfings of the material by the plasma membranes form the endocytosis vesicle. This is a general idea, but if we go into a more detail, then phagocytosis, first thing, if let's say your cells want to take in the bacteria, so this is the bacteria. Okay, so this is our cell. So first thing, if this is our cells, so the cells have to engulf. Okay, now let me draw it out. So first of all, this is bacteria. Then our cells, for example, phagocyte. Okay, our neutrophils, they must extend cytoplasmic projection. Okay, so right now, this is called cytoplasmic projection. Okay, so when this cytoplasmic projection fuse together, try to imagine the cells, so it will form a vesicle. Correct? When they fuse together, this part. Okay, when they join together, fuse together, you're going to form the vesicle. So this vesicle is known as phagocytic vesicle. Are you clear? Okay, so engulf the bacteria inside. Okay, now I have a certain terms that you guys need to understand. First, highlight cytoplasmic projections are formed that extends around the solid particle for phagocytosis, and this projection fuse together, trapping the solid particle within a vesicle. So this vesicle is known as the phagocytic vesicle. Okay. So some books use phagocytotic vesicle, same meaning. Okay. So for example, amoeba capture its prey by extending pseudopodia. I want you guys to highlight here when you use the word pseudopodia, it's only for amoeba. Are you clear? But if you use the leukocytes or phagocytes, now what's the difference between a leukocyte? Leukocyte means white blood cell. Okay, so our white blood cell actually have two major group. One is called phagocytes, another one is called lymphocyte. So phagocyte is the one that can carry out phagocytosis. Okay, who are them? So they are neutrophils and macrophage. We're going to learn this one in immunity, okay, next semester. When they engulf the foreign substances like, for example, bacteria, now neutrophil and macrophages, we use the term cytoplasmic projection. Are you clear? So pseudopodia only for amoeba, but for white blood cells, for example, neutrophil and macrophage, we use the term called cytoplasmic projections. They don't have pseudopodia, understand? Okay, it's not for moving. So pseudo means that's fake one. Podia means the leg. So it's a fake leg for the amoeba to move. But this amoeba also can use this fake leg, okay, to engulf the prey, for example, bacteria. But for our neutrophils and macrophages, they don't have pseudopodia, this structure. So what they have these cytoplasmic projections. So that's why I highlight here. Okay. Now, to play safe, cytoplasmic projection, pseudopodia is also one type of cytoplasmic projection. If you say that amoeba extends cytoplasmic projection, it's still correct. Are you clear? Cytoplasmic projection, as long as something like can extend like this, is known as cytoplasmic projections. Okay. So you can see these diagrams here. Okay. So this amoeba. So amoeba actually extends. You see that amoeba extend the pseudopodium singular. Okay. So you call it, they engulf the food. So guys, why inverted comma here? Because it's not really a food, it's a food particle, okay, or bacteria. So it forms this food vacuole, or I prefer you guys to use the word phagocytic vesicle. Okay, or phagocytotic vesicle. Okay, so can I see that extend and then surround the bacteria? So this is the amoeba. So amoeba, we can use the word pseudopodium or pseudopodia. Okay, but for phagocytes, for example, we do have the macrophages, we do have the neutrophils. Both of them, we have to use the word, okay, cytoplasmic projections. Now, how about pinocytosis? So pinocytosis actually is not extending. You can see that they won't extend. So how they engulf this thing? They engulf it by invaginate. Okay, so something like they form a flask. When they form a flask, you're going to trap this, okay, liquid solutions. Can you see that? They trap the liquid solutions that contain what? Contains the nutrients, for example. So when they take it like this, you're going to form the pinocytotic vesicle or pinocytic vesicle. So basically, they take in the liquid substances. Are you clear? So when they pinch off, they will have this pinocytotic vesicle. If you look at this diagram, can you see that? Okay, so it form a lot of flask-like structures so that when they pinch off, it can form the pinocytotic vesicle. Okay, so for example, the human egg cell take up the nutrient from the surrounding follicles. Okay, and the pinocytosis, the vesicle form actually is smaller compared to the phagocytic vesicle. Okay, so both of them, guys, both of them, phagocytosis as well as pinocytosis, both processes require ATP and involve the take-ins, the components or compound or the bulk material into the cells. So the opposite of the process is known as exocytosis. Okay? Exocytosis. So exocytosis is the reverse process of endocytosis by which the material are removed from the cells. So a vesicle membrane have to incorporate into the plasma membrane and release its contents. For example, plant cells use exocytosis to get the cell wall building material to outside of the plasma membrane. So the process is by exocytosis. You can write down another example, release of the hormone. Okay, from the cells also involve exocytosis. So how you explain for exocytosis? So first we say that's the vesicle that contain the molecule to be secreted. For example, this vesicle like secretory vesicle or Golgi vesicle. So the membrane of vesicle have to fuse. We can use the word fuse or incorporate into the membrane's vesicle. Okay, can I see that? The vesicle, membrane of vesicle fused with the plasma membrane. So when it fused with plasma membrane, can you see that it formed something like a flask and release the chemical or release the material by the process of exocytosis. Okay, I repeat again here. First, your vesicle have to move towards the cell surface membrane. The membranes of the vesicle fuse with the membranes of the cells or plasma membranes, and then release the material by, release the material by exocytosis. And both, guys, look at this, both endocytosis and exocytosis, both of them, okay, do you realize that they need the membrane to be fluidity? Okay, if the membrane don't maintain a certain fluidity, then this process can't take place. You try to imagine sour so rigid. So if the membrane follow the sour so rigid, you won't be able to incorporate. Can I see that? You won't be able to incorporate the membranes into the cell membranes. Are you clear? You won't be able to add or remove. In this case, always remember exocytosis, you add into the membrane. Okay, phagocytosis and pinocytosis, you remove phospholipids from the membrane. So fluidity, only the fluid membranes we can do this. Okay, so with this, I have done for the topics, okay, of the record now.